Hello, my name is Marc de Meyer and I'm an entomologist specialized in the identification, taxonomy and systematics of African flies, in particular Tephritidae fruit flies. I work at the Royal Museum for Central Africa, which is based in Tervuren, Belgium. This presentation is an introduction to the terminology used when describing morphological characters in insects and in particular in fruit flies. It is part of a series developed within the framework of the FFF Fruit Fly Free project, supported by the Standards and Trade Development Facility. In this presentation, I will provide some general background on the general terminology of the insect body parts and focus on the characters specific for fruit flies and often used in identification tools to differentiate taxa. This slide shows the general body structure of a fly, both in dorsal view at the left and lateral view at the right. As any other insect, flies have three main body parts: head, thorax and abdomen. The thorax has a number of appendages, in particular 3 pairs of legs and 1 pair of wings. The latter is unlike most other insects who have 2 pairs of wings. The scientific name for flies, Diptera, refers to this as it stands for the Greek for two-winged. Only the first pair of wings is developed, the second pair being reduced to small knob-like structures that are called halteres. It is also important to understand the orientation of the body and its parts, as identification keys will refer to the relative position of particular structures with regard to each other. Like body part A is placed anterior of part B, or part X is medial while part Y is lateral. Here you see the different orientations. Sometimes orientation references can consist of combinations, like anterodorsal or posteroventral. This is especially the case in structures of the legs on the figure on the right, where the different combinations between A anterior, P posterior, D dorsal and V ventral are given. Now we will look at the different body parts separately. Here we see a drawing of the head in anterolateral view. The head is mainly occupied by the large compound eyes at the lateral sides. Anteriorly, there is a pair of antennae, which are made up of different parts. The antenna itself is carrying a long bristle called an arista. Dorsally, the head has a triangular patch, the ocellar triangle, which is marked by 3 separate eyes or ocelli. Other parts of the head also carry bristles, which all have a separate name. These are usually abbreviated and the full names can be found in any textbook on fly morphology. The most important ones are pointed out here. The frontal bristles, just above the antennae and situated on the fronts. These are pointing forwards. The ocellar bristles, starting from the ocellar triangle, and the orbital bristles between the frontal ones and the ocellar triangle. These are pointing up and backwards. On this slide, you see the positions and development of the head structures in different fruit flies, with the Dacus species on the left and a Cerathites species on the right. Some clear differences can be observed. For example, the last segment of the antenna, the first flagellomere indicated by an arrow and the letter A, is much longer in the Dacus species on the left compared to that in the Cerathites species on the right. The arista, indicated by the letter B, is a bare bristle in Dacus, but has a plumose appearance in Cerathites. Also, the bristles show some differences, with those in Dacus shorter and more poorly developed or absent, like the ocellar bristles indicated by the letter C. You see that these are much longer and strongly developed, including a clear pair of ocellar bristles emerging from the ocellar triangle and pointing forwards. The next slide shows the thorax in dorsal view. In general, one can differentiate three parts. The main part, the scutum, is separated in two subparts through the transverse suture. This allows a distinction between presutural and postsutural areas. Posteriorly, there is a semi-rounded additional structure called the scutellum. Anterolateral, there are two protuberances, the postpronota or shoulders. The thorax is covered by a number of bristles that each have a particular name. We only point out here the most important ones. The postpronotal bristles on the postpronota or shoulders, the dorsocentral bristles in the central part of the postsutural area, the anterior supraalar bristles along the lateral margins and situated above the wings, prescutellar bristles along the posterior part of the scutum but anterior of the scutellum, and finally the scutellar bristles on the actual scutellum. Again, different species can have different thorax shapes, they can be different markings and colors, and different bristles can be present or absent. This is clear from the images shown here. In the following slides, we will demonstrate some particular cases. The slide shows the postpronotal lobe for three different fruit fly species, encircled in red. You can see that the image at the right shows the postpronotal lobe, but there is no bristle present, while the other two images at the left clearly show a dark bristle emerging from this lobe. There's also a difference in coloration, with the one at the left being pale with a dark spot in the middle, the middle image uniformly colored pale, while the image on the right shows a bicolored lobe with darker anterior part and a pale posterior part. In this slide, the presence or absence of the anterior supraalar bristle is shown. This bristle is often easier to observe in a lateral view as in these images. It is present posterior of the transverse suture and dorsal from the base of the wing. The area where to look for this bristle is marked by the red rectangle in the top image. This area is enlarged in the lower images. On the left you see the bristle being present, indicated by the red arrow. On the right it is absent, as no bristle is to be seen in the area that is encircled. Do not confuse this bristle with the notopleural bristle, which is the one seen in the yellow part more to the left and which is present in both species. In the posterior part of the scutum, but before the margin with the scutellum, there can be a pair of bristles present. The area where to look for is indicated by the red rectangular in the top image. In the figure on the left, these bristles are absent, indicated by the areas encircled in red where one would expect them, while on the figure on the right, they are indicated by the red arrows. A general warning: sometimes bristles are broken off and not present anymore. However, there is always a little mark, the antennal implant, on where the bristle used to be. So make sure that there is no such mark if you score the character as being absent. As said, some characters are more easily observed in a lateral view. We already mentioned the presence or absence of the anterior supraalar bristle. Here we see the other main structures that are of diagnostic importance: the postpronotal lobe, the anepisternum, and the cataturgite and anaturgite. The cataturgite and anaturgite are two semi-rounded structures in the posterior part of the thorax and situated next to the altar and below the wing base. In some groups, like day signs, they can be brightly colored yellow. On the left, we see the situation where both of them are yellow. On the right, we see the situation where only the lower structure, the cataturgite, is yellow, while the anaturgite or upper structure has the same brown color as the rest of the thorax. Let us now look at the characteristics of the wing. The wing of a fruit fly shows the normal supporting structure of a network of lines in addition to other marks or bands. All these have particular names and are usually abbreviated. Again, we will only point out the general structure and those of primary importance in identifying fruit flies. We can differentiate lines that run from the base of the wing, the part attached to the thorax, towards the apex, so from left to right in this image. These are called veins. We have highlighted in red a few of these. Each of them has a code. R stands for radius vein, which can split up like seen here in R1 and R2 plus 3. Others are indicated with, for example, an M, which stands for medial vein, or CU, which stands for cubital. These longitudinal veins are sometimes connected to each other with shorter transverse ones called cross veins. These are indicated in red. They are named after the veins that they connect. So for example, the cross vein RM connects the radial vein with the medial vein. BMCU connects the basal part of the medial vein with the cubital vein, and DMCU the distal part of the medial vein with the cubital vein. The area demarcated by veins and cross veins, or between veins, cross veins and the margin of the wing, are called cells. Also, these are given a code and take their names from the veins and cross veins that are bordering them. For example, R1 is bordered by vein R1 and vein 2 plus 3, etc. In addition to this network of veins and cross veins, there are other markings. In the group that are the subject of these presentations, these markings are usually in the shape of bands. In other fruit flies, they can be in the shape of spots. The wing bands each carry a specific name. In representatives of the genus Cerathitis and related genera, we talk of a discal band, an anterior apical band, a posterior apical band, and a subapical band. In addition, there can be some darker spots and streaks in the basal part of the wing. While all species in the genus Cerathitis have a similar shape and appearance, there are some small differences in these bands as shown here on the images. Some are absent, like the posterior apical wing band in the top right image. Some are connected with each other, while others are free. These differences will be used to separate some of the closely related species. In the genera Bactrocera, Dacus and Zogodacus, we have different general pattern of wing bands and a different terminology. Along the anterior margin, there is a dark band called the coastal band. Over the cell BCU, the basal cubital cell, there can also be a darker coloration called the anal streak. Also here we can observe some differences in the general theme. The anal streak can be present like in the image in the top left or absent as the one in the bottom right. The coastal band can be a narrow line like on the bottom right or a broad one expanding like in the top left, or a narrow band which is apically largely expanded into an apical spot like seen at the bottom left. Some extra bands or transverse extensions can also be present like seen in the images on the bottom left or the top right. Finally, there are the legs. There are three pairs of legs, so we can refer to them as either fore, mid or hind leg. As in all other insects, the leg is made up of different components, the most important ones being the femur and the tibia, and to a lesser extent the tarsus. The plural for femur is femora. The most obvious character is the coloration of the femur. It can be uniformly unicolors, figure on the left, or it can be bicolored with one part paler than the other, as the figure on the right. In some ceratid species, we have another typical character which can be used for species differentiation. Some parts of the tibia or the femur can be aligned along the margin with a row of strong and closely adjacent bristles, forming a compact fan of hairs. This is often referred to as feathering because of the resemblance with a bird's feather. We see some examples here of black feathering along part of the tibia and or the femur in some species. The presence of such feathering is a character only observed in male specimens, so there is some sexual dimorphism in these taxa. This concludes our brief and preliminary presentation of the main morphological features used for recognition and differentiation in fruit fly species. Some of these are further explained or illustrated in the other presentations in the set. Many thanks for your attention. This presentation was created in collaboration with Stellenbosch University. We would like to thank the funding agencies as well as the individuals and institutions who in one way or another contributed to the actual development of the presentations.